Good morning, good afternoon, and good evening to other participants around the world. My name is Yunpei. I'm a PhD student from the School of Computer Science and Engineering at Central South University. It's my great pleasure today to present my PhD research titled CellBRF, a feature selection method for single-cell clustering using cell balance and random forest. To begin with the innovative technology of single-cell RNA sequencing, unlike traditional bulk RNA sequencing, which measures the average expression of a large number of cells, single-cell RNA sequencing has emerged as a powerful method for quantifying the transcriptome of individual cells. In single-cell data analysis, clustering plays a crucial role by identifying distinct cell populations, uncovering cellular heterogeneity, and reviewing underlying biological patterns. The steps involved in clustering analysis have been well documented. Generate count matrix, cell filtering, normalization, feature selection, cell similarity calculation, and clustering. Finally, cell types are annotated based on marker genes, enabling downstream analysis such as trajectory analysis. Many factors in these steps affect the clustering performance. Among them, feature selection is an important step that could have a significant impact on the overall clustering performance. Compared to feature extraction, selecting the most discriminative genes can significantly improve the accuracy and computational efficiency and enhance the biological relevance interpretation of clustering results. The motivations are divided into two aspects. First is data imbalance. The wide range of cell types and subtypes can indeed lead to a pronounced imbalance. It's necessary to consider the potential implications of imbalance. The second is that current clustering algorithms mainly rely on a simple method called highly variable genes based on mean variance modeling. Then we have some questions. Using the expression of individual genes for feature selection may not fully reflect the true ability of gene for cell clustering. The clustering of cells is a complex process that involves multiple genes and their interactions. The other is how we determine the number of selected genes. In summary, we require an innovative method that is suitable for imbalanced data and should be capable for optimizing a gene set that accurately reflects the contribution and ability of genes in cell clustering and cell type identification. So we proposed CellBRF. It consists of several steps: data pre-processing, spectral clustering and data balancing, gene importance assessment, and gene subset selection. Similar to previous studies, data processing stage included filtering out lowly expressed genes and performing a log normalization procedure on the gene expression profile. With the pre-processed matrix, CellBRF first predicts the cluster label of each cell by using a spectral clustering flow. Then, we determine the central cluster, minority cluster, and major cluster by using a threshold H based on the number of cells and clusters, C and N respectively. After that, we keep 80% of the cell in each major cluster that have the shortest distance from the cluster centers. And for minority cluster, we use the SMOT algorithm to generate synthetic cells by interpolating between cells and their nearest neighbors. With the obtained cluster labels, the balanced data is then fed into the random forest model to compute the importance of each gene. In the random forest model, Each decision tree is constructed using all cells and a randomly selected subset of genes, and the gene importance is calculated based on the gene index. For example, the importance of feature I on a decision tree is then calculated as follows. Assuming there are only two child nodes. First, we calculate the gene index of node I and child nodes. The importance of node Ni is calculated based on the number of samples Wi and the gene index of node. Then we determine the importance of feature I, Fi, on decision tree using the set of nodes Mi, which split on feature I, and the set of all nodes M. Finally, the overall feature importance is obtained by averaging the importance values from all trees. At last, we select genes whose importance is greater than or equal to the mean plus 3 times the standard deviation. Then, highly redundant genes are removed according to the dynamic threshold. Let's take the example of 4 genes ranked in descending order of importance. We sequentially calculate the linear Pearson correlation between genes. The correlation S12 between the first 2 genes is 0.85. To determine the threshold, we calculate the adjusted value, default is 0.1, divided by the length of the unanalyzed genes plus 1. Add this value to the initial value, default is 0.8, to obtain the final value of 0.82. Since the similarity S12 between the first 2 genes exceeds the threshold, the second gene is removed. After removing these redundant genes, we get the final selected gene subset. We mainly used 4 evaluation criteria to assess performance. Clustering performance is evaluated using NMI and ARI. Higher values indicate better matches with the ground truth label. Additionally, we analyze the distribution of cells in the feature space using silhouette and k-nearest neighbor consistency. Higher silhouette values indicate that the object is well matched to its own cluster and poorly matched to neighboring clusters. K-nearest neighbor consistency is a criterion we define to measure the type consistency of cell and their neighbors. For given gene expression data with C cells, we compute k-nearest neighbors for each cell based on Euclidean distance. Let Fig represent whether cell I and cell G belong to the same cell type, where Fig is 1 if they belong to the same type and 0 otherwise. This indicator calculates the proportion of correct edges in the entire k-nearest neighbor graph, reflecting the degree of consistency between a cell and its k-nearest neighbors. Higher values indicate high consistency between the cell type and its k-nearest neighbors. We obtained 33 single-cell RNA-seq datasets from different sequencing platforms, representing diverse biological scenarios. All data were sourced from various public websites. The results section includes 4 aspects: clustering performance, cell neighborhoods, advantages in CellBRF, and case studies. As shown in the left figure, we compare CellBRF with 6 state-of-the-art feature selection methods: HVG, FIST, gene basis, DUB step, and HRG in terms of ARI on 33 datasets. The right figure is rank distribution of feature selection methods. For each dataset, the methods are ranked from 1 to 7 by their ARI. In summary, CellBRF substantially outperforms all other methods and is the highest ranked algorithm on average across all datasets. If features are well selected, it's expected that in the feature space, the neighbor of each cell can be accurately identified. Based on the known annotated cell type information, we use k-nearest neighbor consistency and silhouette to measure the spatial distribution of different cell types locally and globally in the feature space, respectively. We find that CellBRF achieves better cell type separation on average, indicates that CellBRF can efficiently reduce the distance between cells of the same type while separating cells from different types, thus leading to more accurate reconstruction of cell neighborhoods. To demonstrate the effectiveness of the data balancing strategy, we test the clustering performance of CellBRF in various scenarios: undersampling major cluster, oversampling rare cluster, and without data balancing. Combining with both strategies has better clustering performance than using only one of them, indicating that our balancing strategy is effective in handling imbalanced data that can be used in random forest. The key to ranking-based feature selection methods is whether the adopted gene importance measure can reflect its ability to distinguish cell types or not. Therefore, we compare CellBRF with 4 other ranking-based feature selection methods. We evaluate the clustering performance of each algorithm across a wide range of feature set sizes, from 20 to 4,000, and then calculate the average ARI value across all datasets. As shown in Figure 5a, the genes ranked by CellBRF have remarkable clustering performance. This indicates that the importance calculated by CellBRF can effectively capture the roles of genes in cell clustering. We also compare the clustering performance of the feature sets with different sizes in Figure 5b. CellBRF can identify the optimal feature sets with the highest average clustering performance. To demonstrate the ability to identify cell differentiation stages, we conduct further analysis on a real-time coarse dataset. Xu et al. Performed single-cell RNA-seq at 6 time points along the differentiation protocol to produce definitive endoderm cells from human embryonic stem cells. We use t-SNE to visualize data with the genes selected by CellBRF. Then we identify a total of 3,000 genes with different stage-specific expression patterns and divide the patterns into 3 categories according to the number of genes. We test the clustering performance of genes with different expression patterns. Our results indicate that these genes with real expression patterns may have more discriminative information that can help identify the stages of cell differentiation. Then we calculate the overlap percentage of different expression patterns with respect to the results for each method in Figure C. We further observed that the overlap percentage of genes with real expression patterns in the CellBRF results is higher than those of other methods. To demonstrate the ability to cluster tumor cells, we further analyze the results on the dataset comprising non-malignant cells. The data with selected genes are visualized using t-SNE. For each cell type in this dataset, we collect some marker genes from the PanLawDB database to analyze the composition of the selected genes. We find that the cell type markers occupy the largest proportion in the results of CellBRF, and the cell type marker distribution is relatively balanced, which reflects the advantage of CellBRF on imbalanced dataset. To test the ability to identify cell subtype marker genes, we analyze the expression of CellBRF selected genes in fibroblasts. Interestingly, we find classical markers of myofibroblasts and cancer-associated fibroblasts, including ACTA2, CTGF, and MMP2 in the feature selection results of CellBRF, illustrating the ability of our method to identify cell subtypes. At last, we designed a simulation experiment to evaluate the performance of selected genes in identifying rare cells. We simulate rare cells by reducing the proportion of Joe-cut cells in the data. We replace the feature selection step in a rare cell scoring method, FAIR, with different feature selection methods and calculate the corresponding scores. After sorting cells according to different scores, we count and visualize the changes in the number of real cells on simulated data with 2.5% Joe-cut cells. We can see that almost all cells with high scores are rare Joe-cut cells. We further observe the expression of CellBRF selected genes in the right figure, and we found that these genes are significantly differentially expressed in these two types, including the Joe-cut cell type specific marker CD3D and the 293T cell type specific marker XIST. The main innovation of CellBRF lies in enhancing single-cell clustering performance by combining a novel data balancing strategy with the random forest model and optimizing the feature set according to gene importance and redundant feature removal methods. The performance of CellBRF has been extensively validated on diverse datasets. Source code is publicly available on GitHub. Well, before I finish, I want to thank my supervisor, Professor Jianxin Wang, and Hongdong Li in Central South University, Professor Yaohang Li in Department of Computer Science, Old Dominion University, Professor Jinhui Xu in Department of Computer Science and Engineering, State University of New York at Buffalo. Lastly, I would like to express my sincere gratitude to ISCB for providing the travel fellowship. Their support has been instrumental in making this presentation possible. Thank you all for your attention.